a lot of trying to understand how viruses evolve depends on our understanding of genetics, because essentially that's what they are. They're genetic parasites. They come in, bring in their genome, and then they take over the cell's machinery. So we're going to talk about different viruses today, and sort of hopefully understand how each one has its own genome and its own host. We've talked about tissue tropism. We've talked about host range, and so I don't have the time to talk in a lot of detail. So hopefully you guys can go on to take the virology course, and Dr. Vesinen spends an entire lecture talking about individual viruses. So you'll talk about, HIV individually, he'll talk about flu individually, he'll talk about coronaviruses individually. So a really great course if you're interested in not just the life cycle, but also the immune response and how widespread something might be and things like that. So it's a really great course. Yes, virology. There's virology that's offered I believe every year or maybe every 3 semesters. But it is offered, and it's a great course. So certainly take that if you're interested in learning more about the viruses. So first virus that we're going to talk about is the bacteriophage lambda. So bacteriophage lambda is a temperate virus, right? And it infects E. coli. So E. coli is its host, and E. coli lives in our gut, right? Our colon is where it's naturally found. This is its natural habitat. We are the reservoir for this particular bacterium. And this bacteriophage infects E. coli, right? So it's also found within us. And we know how temperate phage's life cycle works, right? We know that it has these, 2 life style choices. It can either infect its host cell, which is going to be E. coli. We're not the host cell for the phage, so we don't have to worry about it. The phage is a DNA virus, and so it can either enter the lytic cycle after it has infected its host cell. And we know that phages, because of their complex capsid, as well as the complexity of the cell envelope of the bacteria, only inject their DNA into the bacterium, right? So in this case, we're just seeing the DNA being injected. And once the DNA is within the cell, it directs the cellular machinery to synthesize, new virions, and then eventually these cell wall weakens due to production of viral enzymes, and then these cells will lyse, and the new virions can go on and infect brand new cells. So this is our lytic cycle of a bacteriophage. And then you can have the same phage enter the lysogenic cycle, where once the DNA is injected, instead of it controlling these host cell into producing new virions, it gets incorporated into the host genome in what we refer to as a prophage state. And we refer to the cell as the lysogen, I hope you remember. And there were several advantages, but one of the main advantages was that, every time the cell would undergo duplication, the phage will be duplicated as well. And of course, if it's a cell that is metabolically inactive, right, so that it doesn't have a lot of transcription and translation going on, then it makes sense for this prophage to kind of, hide and kind of stick around until cell becomes active, right? And we talked about the process of induction, that if the cell somehow was on path to dying, right? UV exposure being one of such stresses that can be detected on a, as a trigger for death. And so that is a signal for this prophage DNA to excise itself out and then quickly undergo the lytic cycle, right? So that it's a survival strategy for the virus. The bacterium is about to die. If I remain as a prophage, right, there's no further infectious cycle for this virus. this virus needs to be in its virion form. It needs to have all those proteins, as part of its complex capsid so that it could infect the subsequent cell. So, it has to sort of make these decisions, and it's a very complex life cycle for the virus. And like I said, I don't have time to talk about all the details. And there is obviously a lot of signals that are being sensed from the environment, from the host cell as well. We don't necessarily understand how viruses are able to sense that, but obviously for a virus to go from a prophage to undergo induction and excise itself out, it has to be detecting something, right? It has to know that the cell is under stress and it's about to die. So there's a lot that we don't necessarily know. And, so there's a lot that still needs to be learned. But let's today, look at its genome a little bit. And then, how it decides between the lytic and the lysogenic cycle. Again, there are a lot of genes and we don't have time to talk about all of that. So we're going to talk about just a regulatory mechanism that allows this virus to switch from lytic to the lysogenic cycle. So bacteriophage lambda, if you've ever worked in a laboratory and you have used lambda DNA standards, which are used as a marker to determine how far the DNA has traveled on a agarose gel. It's essentially just Hindi 3-treated lambda bacteriophage DNA, which gives you these different size DNA fragments. And then we know exactly what size they are, and we can use them as a standard to measure the size of unknown pieces of DNA fragments. So we use it if you've ever taken any of those DNA courses, like the lab courses, 362, or if you've worked in a laboratory, you're aware that this is used often in genetic engineering labs, wherever biotechnology is involved. So it has a pretty large genome, and it's interesting in that it can either exist as a linear piece, as you could see at the top, or it could become circular and it can exist as a circular piece. So generally within the virions, as it's being packaged into the heads, it would be found in this linear form. But during the lytic cycle, originally when the copying of this DNA begins, you'd remember that bacterial genome is circular, and we talked about how there is this end replication problem, and so you want to avoid that end replication problem. And so the DNA is converted from being circular, from linear into circular, so that the E. coli can easily copy it. And so when we look at the ends of this linear piece of DNA, there are sites that we refer to as cause sites, which stands for cohesive sites. So they are, inverted repeats where they can complementary base pair with each other. And so that is what converts this linear piece into a circular piece. But this happens not while the DNA is within the virion, but rather while this piece of DNA is within the cell, so that the replication process could become easier. So if this piece of DNA, is going to be replicated by the viral, by the bacterial machinery, then it has to come with some sort of an origin of replication that needs to be recognized. So we have initially a early gene product. So when viruses begin to express their genome within the bacteria, you'll have, different genes that are expressed at different times. So you'll hear words like, early genes or early proteins, and then the late genes or late proteins. The early proteins often happen to be, proteins that are expressed by early genes that are involved in the earlier steps of the viral replication cycle. So they might involve things that help with the transcription or replication process for the viral genome. Okay, and when we talk about the late, genes, these are going to be genes which are expressed to, finally package and fully synthesize the virions. So all the capsid genes and the tail genes and things like that, they will be expressed, towards the end of the virus replication cycle or life cycle. So it, depending upon when a protein is needed, generally those that are involved in transcription and replication or controlling the host machinery will be expressed very early on. So we call them early genes, and those that are involved in assembly of virions and their release from the host cell, those will be needed, towards the end of the life cycle of the virus. We refer to them as the late genes. So here we have a gene O product. So this is obviously, an early gene, something that is expressed very early on, and that's going to help initiate the process of replication. And when you look at the replication process, it's a bidirectional replication, something very similar to what we've seen in bacteria, right? So it's going to form that data structure that we talked about. And once the two replication forks run into each other, it's gonna sort of, the replication process ends, but you still have to kind of, detangle the two circular genomes that are joined together. So we've talked about topoisomerase as being involved in that process as well. So, even if things are not mentioned here, I hope you understand that because the host cell is a bacterium, it's limited by, its own enzymes in terms of what it is able to do or what it isn't able to do. Okay, so it is, the phage is using the bacterial host machinery. So, a lot of those same steps are going to be involved, the same proteins are involved, the same enzymes are involved. Okay, but once you have sort of these two genomic DNA resolved from being tangled, the rolling circle method of replication begins. And we've talked about it, that you're going to have an enzyme that comes in to clip, right, one of the strands, and it's generally the outer strand, and the hydroxyl on the 3' end of this nicked DNA is going to act as the primer, which will then be sort of extended, and this extension causes the outer strand to kind of displace itself, okay, as it grows around the inner strand as a template. So inner circular strand is being used as a template, okay, the inner strand is being used as a template. The outer strand is going to use its 3' hydroxyl end to continue to sort of grow. And then as it's doing that, it's going to continue to replace, whatever was there before. And so this process we call the rolling circle method of replication. Hopefully you remember that. And so this is going to continue to replicate the viral DNA, okay? And so you're going to have not a single unit production of DNA, but you're going to have multiple units of phage DNA that are connected together. And so we refer to these as concatemers. And eventually these concatemers will also be converted into a double strand. And you're going to have, as these concatemers are being packaged into the virion heads towards the end of the life cycle of the virus, you're going to have the terminase complex that's going to determine exactly where those cohesive sites are. And it's going to make that staggered cut so that you restore those cohesive sites that you had at the beginning of this life cycle. So although you have several genomic units that are being synthesized together, the terminase enzyme complex is going to ensure that only a single unit is packaged into an individual virion head, which of course we've talked over and over about that, that's such a compact space that it can really just fit that one unit of the genome. So it's not going to fit more than one unit. If you try to fit in two, you're just not going to be able to synthesize, functional virions that are going to go on to be able to infect new host cells, because their synthesis might not even be completed. So this is essentially the replication of the genomic DNA. Remember, replication also refers to the entire life cycle. So if you hear me use it interchangeably, don't get confused. So this is how the genome would be copied. Now, when we look at